I remember that I make a record. I make a record for that class, but I don't know what's happening. Okay, so let me check first. Okay. Can you see my presentation slide?
Yes.
Okay, all right. So let's continue with it. Okay, even though this topic is physiology of pregnancy and lactation, this slide is actually only dedicated for the physiology of pregnancy. Okay, I will share you about the lactation, the slides on the lactation topics afterwards. All right, so let's move on with the physiology of pregnancy. Okay, before this, we did learn on the male reproductive system. Okay, about how does this spermatogenesis occur, and then how does the sperm going to get themselves nurtured? What are the hormones that aids or regulates their maturation process and also the spermatogenesis process? Similar, in female reproductive system, I did mention about the oogenesis process. How does the ovum being produced, and then the being produced and undergo maturations. And I did teach you about the menstruation cycles. Inside the menstruation cycles, we have the menstruation phase, we have the pre-ovulatory phase, by which it's occur in the ovary. All right. So the maturations of the ovum occur in the ovary in one structure, by which the ovum is actually surrounded by the follicle cells, and as they grow up under the influence of the hormonal regulations, okay, upon puberty in females, all right, and then this hormone, um, this hormone is actually telling the follicles to start growing, undergo maturations, change themselves so that they can produce a nutrient to the developing oocyte, okay, and then we did learn that the developing follicles also will produce another type of hormones by which this hormone going to be used up by another system still in the female reproductive system to prepare the female itself so that it can be ready for the developing embryo if fertilizations occur. And then we also have the post-ovulatory phase by which by upon ovulations, what is actually happening in the female reproductive system as a whole, all right, in the ovum itself, in the ovary itself, okay, by which the follicles right now are alone in the ovary. Okay, so what does these follicles doing? By which these follicles right now are known as the what corpus hemorrhagicum at the beginning, and then corpus turn into corpus luteum and regress into corpus albicans if no fertilizations occur. But if let's say the fertilizations occur, so this corpus luteum will still be maintained as the corpus luteum throughout the first trimester of the pregnancy. Okay, so right now we're going to jump on the physiology of the pregnancy. Okay, I did mention that the hormone, the steroid hormone, which is known as the progesterone, together with the estrogens and adenosine hormone, is actually not going to be used by the follicle itself, but it's actually to prepare the female reproductive system so that it can prepare the place for the embryo to develop. Okay, so where is the organ? Where is the organ that this hormone supplied to? It is supplied to the female breast, right, to develop the mammary gland so that later, if let's say the pregnancy occur, they know that the female itself will going to use the breast to feed the baby. All right. So on the other organ, for example, like the changes in the cervical mucus, right? So if pregnancy occur, cervical mucus will become thicker. So let's say why it become thicker, because it's actually act as the protective barrier so that the other microorganism will not pass by. All right, protective barrier, it will form a plug. All right, and then it will not form a plug like what it's happened in animals, but somehow the cervical mucus will become a bit thicker. And then why it's become like that? Okay, refer back to the female reproductive system lecture, by which I did mention that the progesterone together with the estrogens, okay, it actually has an influence in the character of the cervical cervical mucus, okay, towards the ends of the menstrual cycle. So please refer back in that picture. All right, and then about the developments of the uterine cavity itself. So we know that the fertilized egg, okay, will get themselves developed and implant themselves into the uterus. Okay, so if let's say an accident happened and that the fertilized egg does not travel to the uterus, okay, during pregnancy, and it developed in the fallopian tube, because fertilization is actually happening in the upper regions of the fallopian tube, right? So that one we call it as the ectopic pregnancy. Okay, so it's not safe to have implantations at the fallopian tube. Okay, implantations of the baby must occur in the uterus. Okay, let's continue. In this topic, we're going to discuss about the process of the ovum transport. I did mention this in the previous lecture, by which ovum is actually being released into the peritoneal cavity. So how does this ovum being transported into the uterine tube is actually depending on the ciliary movement of the ampulla, right? The infundibulum, okay, and then the muscular contractions of the fallopian tube, and also the movement of the cilia that lining in the fallopian tube itself. So it will aid the movement of the transport. All right. So how does the fertilizations occur, and how does the implantations occur? How long does it take? And then we're going to discuss about the hormones that regulate the regulations of the hormones during the pregnancy. We have progesterone and estrogens, of course, because progesterone and estrogen is coming from the corpus luteum. Please remember, I did mention in the previous lecture, there's still a progesterone and estrogen that being produced by the corpus luteum in the early stage of the pregnancy. Okay, because during that time, the embryo does not develop yet. So they are not able to produce any kind of hormone. So this supply must come from another organism or another organelle or another organ. Okay. And then we're going to discuss about the HCG. Then coming the HCG, HSC, which is hormone chorionic somatomamotrophin, and also other placenta hormone. Okay, and then about the physicals and also physiological changes that associate with the pregnancy in female. Okay, this is the event that occur in the early pregnancy. If you take a look at this one, okay, so ovulation usually we consider as day 0. Okay, so fertilizations will occur in day 1. Right, the ovum is actually available in female reproductive systems only 24 hours. But male sperm can available and live in the female reproductive system around 2 to 3 days. So we can say that sperm shelf life in female reproductive system is actually a lot longer compared to the ovum itself. So ovum will only stay around 24 hours. If let's say the ovulated ovum or ovulated secondary oocyte, I prefer to use it, actually the real term is secondary oocyte, yeah, but I call here ovum because just to spit it out to say like the ovum. So the ovum, like we are going talking right now, is actually the secondary oocyte. Please bear in mind that the one that's being ovulated by the Graafian follicle is actually secondary oocyte or incomplete stage of meiosis to metaphase stage. Okay. Alright, so fertilization we consider as day 1. So the day by which the fetus get themselves alive, we call it as the day 1. And then when fertilizations occur, the fusions of the males and females gamete, okay, it will produce diploid cells. And this diploid cell is actually like a bioreactor. It replicates themselves very, very fast. So during this time, we call it as the blastocyst. So blastocysts, when they form into the blastocyst and they replicate themselves, all right, they will travel into the uterine cavity. So the entrance of the blastocyst that been developed into the uterine cavity took around 4 days, and the implantation is took around 5 days of the ovulation. If let's say you are ovulating, or female is ovulating right now, okay, so the fertilization state the next day around, right? So after 1 week, the implantation is already occurring. Okay. So formation, and then these blastocysts will transform themselves into another type of cells and form and differentiate into a different layer of balls, right? Which called as the trophoblast. And then this trophoblast is actually going to like change in its structure and start to like attach themselves into the endometrium. So it took around day 6, all right? And then on day 8, the trophoblast itself, the what do we call the fertilized egg itself right now in the stage of the blastocyst, okay, by which this blastocyst has differentiated its shell into trophoblast. This trophoblast is actually responsible to produce the HCG hormone, by which if you buy the pregnancy test kit and then you dip that kit into your urine, and then if let's say it's positive, meaning that there is the presence of the HCG. So that HCG is actually coming from the trophoblast, okay? And then the HCG rescue the corpus luteum. So this HCG is not only to be the indicator that you are actually pregnant, but it's actually telling you that not going to be the indicator of the pregnancy, but this HCG is a rescue hormone by which it will rescue the corpus luteum. You will start telling corpus luteum, okay, stay for a while and produce a little bit more of HCG and produce a little bit more of progesterone and estrogen so that the baby can get stronger, get a better place to live on, all right? And then only the placenta will take over and produce the hormone that being produced by the corpus luteum, which are progesterone and also the estrogen, okay? All right, let's move on to the next slide. Okay, this is about the flow charts, how the ovum transport take place. Okay, this is just a recap lecture, so I will not touch this so much. You can read by yourself. Okay, now about ovum transport, I did mention from the previous class that the ovum that being released by the cortex of the ovary, it will be released into the peritoneal cavity. Okay, it will caught by the fimbriae, go through the infundibulum and also reach the ampulla, by which this green region, the fertilizations will occur. And by the movement of the muscle contractions, smooth muscle contractions in the uterine tube together with the fluids movement in the ampulla, ciliary movement of the, it will push the secondary oocytes towards the uterine cavity. Okay, this one, done. The fluid, okay. The fluid, okay. Instead of just talking about the ciliary movement, talking about the, what else? The ciliary movement, there is also the fluid in the abdominal cavity, okay? So that fluid is actually acting like not only a medium to enrich the secondary oocyte, all right, but it also acts as a transportation medium. Okay, so let's move on to the sperm transport. So sperm transportation is actually been divided into 2 portions. Number 1 is the sperm transport in the male reproductive system itself, and the second one, the sperm transport in the female reproductive tract. So meaning that the sperm must be strong enough so that it can pass into 2 phases, by which in the male itself and the second one in the female reproductive tract. Okay. All right. So in the sperm transport, I did mention this in male reproductive class already, but we just have a quick recap on this. So spermatozoa actually will be transported via the testicular fluids. All right. So this from the testes, it will move to the seminiferous tubules. All right. Through the seminiferous, the coil tubules inside the testes itself, and then pass through the vas deferens, recta testis, and the caput, and then go to the head of the epididymis. And then in this one, they have a seminiferous tubule. They have a fluid, by which this fluid is actually will act as the transportation system that flood the sperm together with the movement of the smooth muscles in the epididymis, all right, and also the ciliary current inside the epididymis itself. It will move the sperm into the efferent ductus. Okay, so within 12 days, all right, so the sperm that being released will undergo the biochemical maturations. So please remember the spermiogenesis process. Okay, so during this time, the spermiogenesis process is occurring, by which in the plasma, by which during the maturations, during the spermiogenesis, the sperm will undergo maturations, by which it will form the it will like arrange or arm themselves to have the acrosomal sections at the sperm head, and then all the mitochondria will move to the neck, and then they start forming the tail that get themselves ready so that they can swim and pass through the male reproductive system out of the male reproductive system. Okay, so once the ejaculation occurs, so what is ejaculation? Ejaculation is actually rapid pass. Rapid pass through what? Through the ductus deferens. So from the ductus deferens, this sperm will mix with the fluid, okay, together with the seminal vesicles, together with the prostate gland fluid, together with the bulbourethral gland fluid. Okay, all of this will not actually going to be the transportations or medium for the sperm to be transported out, but it actually will nourish and give the environment that is conducive for the sperm to carry out its function, okay, by which to swim so that it can reach to the destination. Alright, so prostate gland, recap back, it is actually more acidic, okay, in the seminal vesicle it's much more alkaline. So when the prostate gland fluid mix together with the seminal vesicle fluid, it will become more natural. So the natural pH of the surrounding of the medium is actually give the sperm give the sperm much more energy. Okay, it will produce the correct pH environment that is much more convenient to the sperm to make a move. Because at the beginning, the sperm are not able, even though it has a tail, it actually does not have an energy to move. But by using the by the changes in the pH of the environment, it then gain its ability to make a move, become much more motile. Okay, and then let's take a look at this seminal fluid. So this seminal fluid is actually will be deposited together with the sperm in the upper vagina, right? So just imagine that this is the upper vagina. Okay, look at this illustration, and then the compositions of the seminal fluid, all of this together with the acidic environment of the vagina, it will actually protect the spermatozoa. Okay. Look at the pH. We'll see that the pH of the vagina is actually 4.3. That's what we discussed in the class before. And then the pH of the seminal fluid is quite alkaline. And then when it mix together, it will form the pH around 7.2. So 7.2 is actually optimal for the sperm motility. Okay, so talking about the cervical kyna and cervical mucus. So as I mentioned, at the end of or during ovulations, alright, the female will have high level of progesterone and estrogen, by which this progesterone and estrogen will change the characteristic of the cervical mucus. So the characteristic of the cervical mucus is actually during ovulation, is actually slippery. Okay, do you still remember? So that one will be a very conducive environment for the sperm to pass through. If let's say other than the ovulation period, the cervical mucus will become thicker. Alright, and another point in here is that the vagina itself, okay, the environment, the structure of the vagina itself, once the sexual intercourse occur, alright, so it will create the intravaginal pressure. So it will create a suction force. So this suction force inside the vagina actually become one of the mechanism that assists the movement of the sperm into the female reproductive structure. Okay, instead of the swimming movement of the sperm itself lah. Okay. Alright, this one I did mention. Done. Okay, so there are two modes of transport of the sperm in the cervix itself. Right, so just now we talked about the intravaginal pressure. Right now we move to the movement of the sperm in the cervix. So there are two, which is the first one is the initial rapid transport. Number two, by the swimming of the spermatozoa. Okay, so in the cervical, in the female cervical tract, right, cervical region, it actually consists of a muscle. So this muscle will help the sperm to pass through. Okay, looking at this, it's only 5 to 20 minutes to reach the uterus. Alright, so swimming of the spermatozoa, of course, because during this time, as the environment, as I mentioned earlier, by which the pH is already become much more neutral comparing to the original one. So, okay, wait for a while. I have a problem with my washing machine. Okay, let's continue. Okay, we stop at the pH. So that pH is actually is conducive for the sperm to start moving and swimming. So look at the travel rate. It's only around 2 to 3 millimeter per hour. So the swimming motions of the sperm is actually not as what we imagine. If you look at the video, sometimes you can see like, oh, so fast. Actually, it's not. Okay, but that's why it needs a help from the movement, the muscular contractions of the cervix. So if we depends only on the swimming motions of the sperm itself, okay, without the contractions of the female reproductive structure, it will take 2 to 4 days. So during this time, it's 4 days the sperm already died. Okay. And then the passage of the spermatozoa through uterine cavity. Yes, it must pass uterine cavity because sperm will not fertilize the egg in the uterine cavity. It needs to go first to the uterine tube, okay, mating over there and then come back to the uterus. Okay, oh, so far the journey takes. Alright, so the contractions of the uterine smooth muscle also is actually, I did mention, actually much more have a greater impact compared to the sperm motility. Okay, this one. Alright, so now let's imagine, let's imagine that we have a million of sperms actually penetrating through the uterine cavity, okay, and then start swimming and competing with each other, okay, through uterine cavity and go to both left and side of the uterine tube. But usually, the sperm also clever because they have a chemoattractant to the ovum. So usually it will go to the way by which the ovum being released. If the ovum is there, the secondary oocyte is there, they know where to go. They will not go to the other region that doesn't have the secondary oocyte because our ovary actually will release one egg at the right or left, okay, alternately. So maybe this month is on the right side, next month on the left side. So if let's say that this month is the secondary oocyte is coming from the right side, all right, so the sperm will start competing with each other and swimming toward the right side of the fallopian tube. Okay, so when they reach into the fallopian tube, since I mentioned that they take 2 to 3 days, okay, their shelf life is 2 to 3 days, they actually like will embed themselves in the cilia of the uterine tube of the female reproductive tract. Why they stay in the cilia? Why there are a lot of sperm that actually only one will penetrate the secondary oocyte, but the remaining will get themselves shed at the cilia, at the epithelium of the isthmus. Okay, what are they doing over there? It's actually to create a sperm reservoir. Please remember that not all the sperm, even though it reached to the line, okay, reach to the target, it can still alive. Sometimes it's die towards the end. So that's why we need a sperm reservoir. So that region will become the regions for the sperm reservoir. All right. Okay, during shading region, okay, by which they stay at the R&R. Okay, I said the what do we call the isthmus itself as the R&R because the sperm will be collected over there. Okay, will stop or take rest over there. What does the sperm doing? So the sperm is actually not take rest, but is actually preparing themselves furthermore so that it can get ready to penetrate the secondary oocyte. Like take a lot of energy, like take all the nutrients, undergo the capacitation reaction so that it can get themselves ready, arm themselves to penetrate the secondary oocyte. Okay, so that process is known as the capacitation process. So in the capacitation process, okay, it's actually initiated, it started when the sperm pass through the cervix and it will complete in the fallopian tube. So once the sperms pass through the female reproductive tract, through the cervical, to the trunk cavity, go to the fallopian tube, during this time the capacitation process is actually occurring. Alright, so what is actually happening is that they will undergo the acrosomal reaction. Okay, we're going to take a look at this one by one later on, and then it will bind to the zona pellucida. So what is zona pellucida? The region that shed the secondary oocyte. Please refer back. In the secondary oocyte, we have a region, okay, which the secondary oocyte at the outer layer is the zona pellucida and the outermost layer is the corona radiata. Still remember? So that corona radiata, the crown that radiates. Still remember? So the ovulated egg, the secondary oocyte that has been ovulated has the corona radiata, zona pellucida. Okay? And then during this time, they will start to acquire their hyperactivated motility. So during when? During the capacitation process. Alright, so this illustration going to show you on the, sorry, I have to go a bit fast because I have a lot to tell you on this lecture. Alright, so in this one, okay, we have the illustrations on the capacitation process. So this one is in the epididymis. So in the epididymis, on top of the sperm head, they are actually have a what we call a surface protein. So this surface protein is made up of the protein and carbohydrate. So we call it as a glycoprotein structure. Okay, all right. And then once it has been ejaculated, okay, so on top of the sperm head, okay, because before they has been ejaculated, they will be flat into the seminal fluid. Inside the seminal vesicle fluid, they have a factor that will arm the sperm head. So that factor is known as the decapacitation factor. So the seminal vesicle fluid, they consist of protein that will attach or bind to the sperm head once the sperm pass into the seminal vesicles. Into the seminal glands. Right. And then we have the capacitation process. After the capacitation process, you will see that the head of the sperm is right now being exposed. So we can say that actually before the sperm has been released out, they have the arm or a shield that will protect the nucleus inside the sperm head. So when during capacitation process occur, alright, so all the glycoprotein will be removed. Alright, so right now when it's all removed, these head are actually penetrable for the secondary oocyte. Alright. Okay, did mention to you. Alright. I did mention this to you also. Okay, done. Okay, done. Okay, done also. Yeah. Okay, all of the wording. Okay, why did I did not mention to you all of this? Because I did mention while I'm talking. So you can read this as your reference. Alright, so now let's move on to the fertilization. So we know that already being transported into the ampulla of the uterine tube, and the sperm right now has passed through the first stage by which to move through the male reproductive system and pass through the second stage, move via the female reproductive tract, undergo the capacitation process, decapacitation process, and start pulling themselves in the uterine tube, okay, so that they can get themselves ready. All right, during this time, the capacitation process is continue, and then they start to reach the secondary oocyte. Okay, remember that the movement is not only via the motility of the sperm itself, but it's being helped by the muscular contractions of the female reproductive tract. Okay, so the fertilization is actually the fusions of the male and female gametes. So usually it takes around 24 hours after ovulation. If let's say I buy the ovulation test kit, okay? Remember, pregnancy test kit is different from the ovulation test kit. So I try to conceive, okay? I try to have a baby. Let's say I'm trying to get pregnant, and then I bought the ovulation test kit. And then this test kit I did this test into my urine, and then I start noticing that there are a lot of the LH hormone. There is a positive sign by which the presence of the LH hormone. And then I and then after 24 hours, the ovulation start occurring, and then during 24 hours, okay, so if fertilizations occur, if I go dating with my husband, for example, okay, and then fertilizations occur within 24 hours. If let's say I didn't go, so within 2 weeks, the secondary oocyte will be flushed out together with the menstruation periods. All right, so the fertilization will take place in the ampulla, please remember. Okay, and then the movement is via peristalsis, contractions of the uterine muscles, and instead of just having that, in females, we have the oxytocin. So oxytocin is actually helping the what do we call the female reproductive structure muscles to contract. Okay, so especially this oxytocin will be released during sexual intercourse. Alright, instead of that, we also have the prostaglandin. So prostaglandin not only present in the high concentrations of progesterone, okay, it's also been present during in the seminal fluid. So please remember back, what does the prostaglandin do? Prostaglandin will trigger the muscular contraction. Okay, so I did mention just now, how does the sperm know that the secondary oocyte is there? It's via chemo attraction. On top of the on top of the secondary oocyte, there is a receptor that is specific with the receptor with the protein on top of the sperm. Okay, so the sperm can know where they need to go. So via the chemo attraction process. All right, and then number 2, if you take a look at this, is actually the process of the adherence of the sperms on the zona pellucida. Okay, this is the secondary oocyte. Outside of the secondary oocyte, we have the zona pellucida. So at the beginning, the sperm will bind to the regions known as the zona pellucida first, and then the penetration will start occurring where the acrosomal reactions. Once they bind, okay, the protein bind to the receptor. It will change the chemical structures of the protein, all right? And then the acrosomal fluid on top of the sperm head will start leaking, okay? So this acrosomal fluid, this one called as the acrosomal reaction. So what is the importance of this acrosomal fluid? It's actually like a peroxisome. So it will hydrolyze the secondary oocyte membrane so that it can pass through. Right, so and then the adherence and then the pass through of the nucleatic content of the sperm into the egg cytoplasm. Alright, so please remember on the secondary oocyte that being released, they have the corona radiata, they have the zona pellucida, okay, the region over here in between the zona pellucida and also the membrane of the secondary oocyte known as the perivitelline space, okay, and the membrane itself is known as the vitelline space. Oops. Okay. As a whole, okay, if you take a look at this, the sperm is actually will move, okay, past the follicular cells and bind to the zona pellucida again. All right. Remember that on top of the zona pellucida, there is a receptor for the protein, okay, the capacitation protein on top of the sperm head just now. Okay, and then once the sperm had bind to the zona pellucida, okay, so the chemical reactions occur. Okay, this chemical reaction will start increase the calcium concentrations on top of the, while it binds to the receptor, it will increase the calcium concentrations inside the sperm cytoplasm. So when the concentrations of the calcium in the sperm cytoplasm increase, okay, the acrosomal reactions will occur. So it will release the hydrolytic enzyme, and this hydrolytic enzyme at the acrosomal head of the sperm. Okay, this one we call as the acrosomal reaction, yeah. Okay, so this hydrolytic enzyme will start hydrolyze the zona pellucida. So this zona pellucida is actually acting like a barrier. So this hydrolytic enzyme will digest that barrier so that it can reach the perivitelline space and fuse with the vitelline region. Okay, so we're done with this, and then we take a look at this one. Once the sperm head bind or reach the perivitelline space, it will increase the calcium concentration. It will start activate the secondary oocyte to undergo the influx of the calcium. So once the calcium influx occur in the secondary oocyte, okay, just now in the sperm cytoplasm. Right now, when the secondary oocyte has been intruded by the sperm head, okay, via the acrosomal reaction, the calcium concentrations inside the secondary oocyte also will increase. Okay, so what does this calcium cause, or what does this calcium trigger? So this calcium is actually will cause hardening of the perivitelline region. So when the hardening of the perivitelline region occur, it will block the other sperm from coming in. So this is only one sperm, okay, that can pass through. Right now, there is actually not only one that trying to fertilize the egg. There are millions that trying to fertilize the egg. But logically thinking, how does the secondary oocyte accept only one but not two, sometimes two, but why not 3, 4, 5, 6? Sometimes there are 5, okay, very rare. All right, why? What are the mechanisms that make the secondary oocyte know that I will only allow only one sperm or two sperm only to get in? Two also are quite rare but common. Okay, 5 is very rare. Okay, so that one we call as the block of polyspermy. Okay, polyspermy means many sperm. So the secondary oocyte have the capability to block polyspermy. So how does the process occur? Is via the increase in the calcium concentrations in the egg cytoplasms. Alright, so when increase in the calcium concentrations in the egg cytoplasm, they start to cause hardening, okay, hardening of the vitelline space. When the hardening occur, there will be no other sperms can bind to. And then when the sperm are capable to fuse with the egg cytoplasm, okay, it will release the genetic material, okay, the haploid genetic material into the secondary oocyte cytoplasm. But please bear in mind that in a sperm, there has the neck and also the tail. So the one that being released, the one that being fused with the secondary oocyte is only the head. The tail will left behind. Because please remember that what is the function of the tail? The tail is to assist the movement. So once it reaches into the movement, you will no longer need the tail. So tail, goodbye. Alright. So only genetic materials will be passed into the secondary oocyte, and then the fusion of the nucleus will occur. Right. There you go. The process of development of the fetus will start occurring. Okay. This is the whole content. You can read by yourself. Okay. I just tell you by using this image so that you can see in the sequence. Okay. This one is just an enlarged version. You can read for. I put it over here because I'm afraid that you cannot read from this small diagram. All right. So in the acrosomal reaction, let's recap back. In the acrosomal reaction, there are actually the breakdown of the lysosome will release 2 enzymes. So what are they? They are acrosin and also ferlin. So acrosin is actually like the one that is required to penetrate or digest the zona pellucida, and the ferlin is actually very crucial so that it can ensure that the sperm nucleus going to fuse with the sperm with the ovum membrane. Okay, the sperm membrane are capable. I mentioned nucleus just now. The sperm membrane can fuse with the ovum membrane. All right. So, all right. In the cortical reaction, okay. In the cortical reaction, okay, in the cortex of the secondary oocyte, okay, that one we call as the cortical reaction. Okay, sini ya. Okay, this is acrosomal reaction. When it reach the cortex, it will start undergo cortical reaction. So in the cortical reaction, okay, sorry. Okay, in the cortical reactions, once the male the sperm fuse with the female secondary oocyte, it will increase in the calcium concentrations in the egg cytosol. Okay, what actually is happening? How during this cortical reaction, it the block of the polyspermy is actually occurring. So meaning that cortical reaction is actually the process to block polyspermy. So how does the blockage of the polyspermy occur? The first one, there are a step. The first one is the swelling of the perivitelline space. The second one, the vitelline layer will start hardening. And the third one, it will clipping the sperm binding receptor. So how does that occur? Okay, once the sperms undergo its acrosomal reaction and it reach, over here, sorry. Okay, so once the sperm bind to the pass through the zona pellucida, bind to the pass and meet the perivitelline space, okay, later to the vitelline layer, these acrosomal reactions will causes the influx of the genetic materials of the male into the egg cytoplasm. Okay, once this occur, the cortical reaction will take place. So during the cortical reactions, you will see that the receptor, okay, this is a cortical reaction, yeah. This is the receptor for the sperm to bind. So the space, the perivitelline space, this is a perivitelline space. The perivitelline space will start undergo swelling. So when it swell, it will burst out this layer and it will clip off this receptor. So what does this receptor? This receptor is actually the receptor for the sperms to bind. We call it as the sperm binding receptor. So once this one has burst out, the receptor also has been removed. So there will be no sperms can bind to the fertilized secondary oocyte. So that's how the block of the polyspermy or the cortical reactions is occur. Okay, so done telling about this. Is just a summary of the process that I told you just now. You can read this by yourself. It's just for your reference. All right, now once the fertilizations occur, there you go. There will be a day one of the day one of the baby's life. All right. So the fertilized secondary oocyte, we call it as the conceptious. Okay. So once it fertilized, the replication process is occurring like crazy. Okay. So you will see it will undergo mitosis and form 2 cells, and then 4 cells, and then 8 cells, and then become 16 cells. The stage of the 16 cell, we call it as the morula. All right. So okay. All right. In the what do we call this one, morula? This morula is actually can stay in the uterine tube for 3 to 4 days. Okay. So right. So this is the whole the illustrations on the formations of the morula. Fertilized egg, okay, you will become a diploid cell right now, and then undergo mitosis to form 2 cells, 4, 8, 16. The 16th stage we call it as the morula, and then it will form into 32, 64. This one known as the blastocyst. So during this time, these cells is actually totipotent. So what does that mean by totipotent? So totipotent is actually the cell itself. If you take one cell out of it, okay, you take one cell out of it and then you culture in a culture plate, it can form one individual. Okay, it can clone to another baby. All right, a clone with another baby with the same genetic code lah. All right, so meaning that one is totipotent. So totipotent meaning that it can develop and differentiate into another organism. Meaning that if let's say this is you, okay, once upon a time, okay, all right, during your early 1 day stage, if I take one cell out of the blastocyst, one cell out of the morula, I put on a culture dish and I clone and I culture it, and later it will grow up to be you. Contohnya Patrick. Dan satu hari, one day I will get Patrick out of the morula. That's how they clone the dolly, the what do we call? Apa? Shape. Dolly shape. If you learn in genetic lah. Okay. So if let's say that this blastocyst has differentiate cleave into two, so it will form the identical twin. Okay. Alright. So if you take a look at this morula, okay, this morula is actually still have the zona pellucida that protect themselves during this time. So why does the zona pellucida is still there? That because the presence of the zona pellucida will prevent this morula to bind to the uterine tube. Okay, so if the morula is if the zona pellucida is shut off, it is capable to undergo implantation in the uterine tube. That's why in the uterine tube, there's still the zona pellucida covering the fertilized egg. Okay, first one, prevent adhesion, and then it will protect the conceptions from the mechanical damage. Why mechanical damage? Because during this time, the uterine tube is contracting. So it gives mechanical forces to the replicating morula. So this zona pellucida will protect the developed morula. Alright, and then the next one is due to the prevent the early immunological rejection. So immunological rejection is because it does not fuse with the mother yet during this time. So the body will start detecting that one will be the intruders, okay, other organism inside. So that's why the zona pellucida is protecting it. Okay, blasting. Okay, later on this morula will form into 16 cells, will become blastocyst. Alright, this is a fertilization, this is implantation. This one we call it as the ectopic pregnancy. So implantations that occur out of the uterine cavity. Okay, upon 7 days of the ovulation, you will see that the corpus luteum will still secrete the progesterone. Okay, so this progesterone and estrogen will start preparing the uterine cavity to make a bed for the blastocyst to implant themselves. Implantation, okay, after 21 days. So after 21 days of menstruation cycles, meaning that 7 days of ovulation. So after 7 days of ovulation, it start moving to the uterine cavity. This blastocyst will start move to the uterine cavity, and this blastocyst will differentiate into trophoblast, and then this trophoblast will get themselves buried into the uterine cavity. Okay, so in the blastocyst, which I said they are in the beginning they are totipotent. Okay, once they move themselves into the uterine cavity, they will start to differentiate into 3 different types of cells. So number one, we call as the inner cell mass. So this inner cell mass, the blue color one over here, will become the baby. All right. So the brown color, the cells that lining that form like a ball over here is the trophoblast. This trophoblast will actually differentiate into the one will produce like a finger-like structure that make themselves capable to implant into the mother's or uterine's womb. All right. And then we have the blastocyst cavity. This one will form the amniotic fluid. Okay, this one. All right, I did mention to you this one. Okay, let's take a look at the trophoblast. Okay, just now I did mention that the trophoblast will form like a projection or a finger-like structure, differentiate into a finger-like structure, so that it can implant themselves into the uterine cavity. Instead of that, this trophoblast also will differentiate into a cell that is capable to give a nutrient to the inner cell mass, to the developing embryo. And it also will secrete the HCG hormone. So if let's say the question is coming out like, what are the cells in the blastocyst that that are responsible to produce the hormone, the first hormone that detect the pregnancy, which is the HCG, it is the trophoblast. All right. Okay, I did mention this to you. Okay, now let's take a look at this one. I go fast because this is just what the same thing which I mentioned to you earlier. Okay. Alright, so on the 8th day of the ovulation, okay, just now on 7th day, it start to embed themselves in the uterine cavity. On the 8th day, okay, it embedded into the endometrial stroma. Alright, so this trophoblast just now, this one, this is trophoblast, the green in color. This is the inner cell mass, all right? So this is the trophoblast. So this trophoblast will differentiate into one. So number one is the two type of cells. Okay, the pale green color over here is the cytotrophoblast, and the uh, dark green color over here is the syncytiotrophoblast. So the cytotrophoblast is actually are capable to undergo mitosis and form a protective layer all around the developing embryo. And this syncytiotrophoblast is actually cannot really regenerate on its own. It needs help from another region. And if you take a look at this, you will see that this syncytiotrophoblast will form a vacuole-like structure over here. So this vacuole-like structure, we call it as the lacunae. Okay, so what is the importance of the lacunae? So this lacunae is actually the region by which the blood vessel will form. Okay, so this blood vessel will start forming, and this blood vessel later on will form the umbilical cord. So remember that in the umbilical cord, there is a fusion of two bloods, which is from the mother's blood and also the baby's blood. You will see, okay, this is the cytotrophoblast. This is the syncytiotrophoblast. This syncytiotrophoblast later, okay, this is the uterine wall, okay. So this syncytiotrophoblast later will start fuse and form the endometrial membrane. It will fuse with the mother's endometrial layer. So maternal endometrial. Okay. There you go. You will see that the trophoblastic lacuna will start forming a blood vessels. It start forming, all right, and then the fuse to form the lacuna contains the maternal blood and also the debris from the uterine gland. All right. It will also this fluid we call it as the embryo trough. Okay. So this fluid is actually the one that will supply the nutrients to the embryo. Because bear in mind, even though the cells that develop itself, they can get nutrient by themselves, but as they grow big, they need a support. So the nutritional support for the developing embryo is actually coming from the maternal circulations. That's why the pregnant lady should eat a lot of nutritious food, so that all the foods that coming into the mother's blood will be supplied to the developing embryo. Remember that the developing embryo is actually like a bioreactor. It replicates very, very fast. So female is actually very good lah. Right. Can produce another person with the help of male. Okay. So this syncytiotrophoblast later will secrete the HCG. So remember back, syncytiotrophoblast is actually coming from trophoblast. Okay. Trophoblast will differentiate into two structure, which is cytotrophoblast and also syncytiotrophoblast. So syncytiotrophoblast will produce HCG. Okay, so it will release the HCG in the lacunae. So what is in the lacunae is actually the blood. So HCG will be released in the blood, and that blood will be transported and fused together, combined together with the maternal's blood. That's why you will detect the HCG in the maternal blood. Okay, so instead of that, this HCG also actually will act to protect the corpus luteum. So at the beginning, HCG is actually a rescue for the corpus luteum. It tell the corpus luteum, please don't die yet. Please produce a lot more of progesterone and estrogen. I still need your help until I get myself ready. Okay, so that's for the pregnancy and implantation. Now let's take a look at the hormone in the pregnancy. So what are they? Beforehand, it's 10 o'clock already. Do you have class after this?
Yes.
Yes. At what time? 10. Oh, exactly at 10. Oh, yes. If that's so, we can. I think we can continue this one for the coming class lah. Okay. Beforehand, please help me to go to your chat box. Help me to fill in your attendance in the Google Sheet. And I think I'm going to continue on the roles on the hormone of pregnancy it's just about to end. But since you have class at 10, I think I don't want to take others' time. I will continue together with the lactations and also the menopause. Okay, lactation and menopause is just about a quarter. Okay, please help me to fill in the Google form, yeah. Uh, okay, since you have class at 10, I think this marks the end of our lecture today. I will continue for the rest of the topics on the hormone pregnancy and what for the changes in the pregnancy. It's not so much. I feel like to go in the next class. Okay, I think that's all. Thank you very much for joining in, and please don't forget to fill in your Google form. Okay, so you may dismiss the class. Thank you.
Thank you. Thank you, Doctor.
You're welcome.